15.5 Ribosomes and protein synthesis. By the end of this section, you will be able to do the following. Describe the different steps in protein synthesis. Discuss the role of ribosomes in protein synthesis. The synthesis of proteins consumes more of a cell's energy than any other metabolic process. In turn, proteins account for more mass than any other component of living organisms, with the exception of water, and proteins perform virtually every function of a cell. The process of translation, or protein synthesis, involves the decoding of an mRNA message into a polypeptide product. Amino acids are covalently strung together by interlinking peptide bonds in lengths ranging from approximately 50 to more 15.5. Ribosomes and protein synthesis 383 than 1000 amino acid residues. Each individual amino acid has an amino group, NH2, and a carboxyl, COOH, group. Polypeptides are formed when the amino group of one amino acid forms an amide, i.e., peptide, bond with the carboxyl group of another amino acid, figure 15.15. This reaction is catalyzed by ribosomes and generates one water molecule. Figure 15.15. A peptide bond links the carboxyl end of one amino acid with the amino end of another, producing one water molecule during the process. For simplicity in this image, only the functional groups involved in the peptide bond are shown. The R and R designations refer to the rest of each amino acid structure. The protein synthesis machinery. In addition to the mRNA template, many molecules and macromolecules contribute to the process of translation. The composition of each component may vary across species. For example, ribosomes may consist of different numbers of rRNAs and polypeptides depending on the organism. However, the general structures and functions of the protein synthesis machinery are comparable from bacteria to human cells. Translation requires the input of an mRNA template, ribosomes, tRNAs, and various enzymatic factors. Note: a ribosome can be thought of as an enzyme whose amino acid binding sites are specified by mRNA. Link to learning. Click through the steps of this PBS Interactive. http://openstax.org/l/prokaryote_protein to see protein synthesis in action. Ribosomes. Even before an mRNA is translated, a cell must invest energy to build each of its ribosomes. In E. coli, there are between 10,000 and 70,000 ribosomes present in each cell at any given time. A ribosome is a complex macromolecule composed of structural and catalytic rRNAs, and many distinct polypeptides. In eukaryotes, the nucleolus is completely specialized for the synthesis and assembly of rRNAs. Ribosomes exist in the cytoplasm of prokaryotes and in the cytoplasm and rough endoplasmic reticulum of eukaryotes. Mitochondria and chloroplasts also have their own ribosomes in the matrix and stroma, which look more similar to prokaryotic ribosomes, and have similar drug sensitivities, than the ribosomes just outside their outer membranes in the cytoplasm. Ribosomes dissociate into large and small subunits when they are not synthesizing proteins and reassociate during the initiation of translation. In E. coli, the small subunit is described as 30S, and the large subunit as 50S, for a total of 70S. Recall that Svedberg units are not additive. Mammalian ribosomes have a small 40S subunit and a large 60S subunit, for a total of 80S. The small subunit is responsible for binding the mRNA template, whereas the large subunit sequentially binds tRNAs. Each mRNA molecule is simultaneously translated by many ribosomes, all synthesizing protein in the same direction, reading the mRNA from 5' to 3' and synthesizing the polypeptide from the N-terminus to the C-terminus. The complete mRNA, polyribosome structure is called a polysome. tRNAs. The tRNAs are structural RNA molecules that were transcribed from genes by RNA polymerase III. Depending on the species, 40 to 60 types of tRNAs exist in the cytoplasm. Transfer RNAs serve as adapter molecules. Each tRNA carries a specific amino acid and recognizes one or more of the mRNA codons that define the order of amino acids in a protein. Aminoacyl tRNAs bind to the ribosome and add the corresponding amino acid to the polypeptide chain. Therefore, tRNAs are the molecules that actually 384 Chapter 15, Genes and Proteins access for free at openstax.org. Translate the language of RNA into the language of proteins. Of the 64 possible mRNA codons, or triplet combinations of A, U, G, and C, 3 specify the termination of protein synthesis and 61 specify the addition of amino acids to the polypeptide chain. Of these 61, one codon, August, also encodes the initiation of translation. Each tRNA anticodon can base pair with one or more of the mRNA codons for its amino acid. For instance, if the sequence KUA occurred on an mRNA template in the proper reading frame, it would bind a leucine tRNA expressing the complementary sequence, GAU. The ability of some tRNAs to match more than one codon is what gives the genetic code its blocky structure. As the adapter molecules of translation, it is surprising that tRNAs can fit so much specificity into such a small package. Consider that tRNAs need to interact with 3 factors. 1. They must be recognized by the correct aminoacyl synthetase, see below. 2. They must be recognized by ribosomes. And 3. They must bind to the correct sequence in mRNA. Aminoacyl tRNA synthetases. The process of pre-tRNA synthesis by RNA polymerase III only creates the RNA portion of the adapter molecule. The corresponding amino acid must be added later, once the tRNA is processed and exported to the cytoplasm. Through the process of tRNA charging, each tRNA molecule is linked to its correct amino acid by one of a group of enzymes called aminoacyl tRNA synthetases. At least one type of aminoacyl tRNA synthetase exists for each of the 20 amino acids. The exact number of aminoacyl tRNA synthetases varies by species. These enzymes first bind and hydrolyze ATP to catalyze a high-energy bond between an amino acid and adenosine monophosphate, AMP. A pyrophosphate molecule is expelled in this reaction. The activated amino acid is then transferred to the tRNA, and AMP is released. The term charging is appropriate, since the high-energy bond that attaches an amino acid to its tRNA is later used to drive the formation of the peptide bond. Each tRNA is named for its amino acid. The mechanism of protein synthesis. As with mRNA synthesis, protein synthesis can be divided into 3 phases: initiation, elongation, and termination. The process of translation is similar in prokaryotes and eukaryotes. Here we'll explore how translation occurs in E. coli, a representative prokaryote, and specify any differences between prokaryotic and eukaryotic translation. Initiation of translation. Protein synthesis begins with the formation of an initiation complex. In E. coli, this complex involves the small 30S ribosome, the mRNA template, 3 initiation factors, IFs, IF1, IF2, and IF3, and a special initiator tRNA, called tRNA-METF. In E. coli mRNA, a sequence upstream of the first August codon, called the Shine-Dalgarno sequence, AGG-AGG, interacts with the rRNA molecules that compose the ribosome. This interaction anchors the 30S ribosomal subunit at the correct location on the mRNA template. Guanosine triphosphate, GTP, which is a purine nucleotide triphosphate, acts as an energy source during translation, both at the start of elongation and during the ribosome's translocation. Binding of the mRNA to the 30S ribosome also requires IF3. The initiator tRNA then interacts with the start codon August, or rarely, GUG. This tRNA carries the amino acid methionine, which is formulated after its attachment to the tRNA. The formulation creates a PHO peptide bond between the formal carboxyl group and the amino group of the methionine. Binding of the F-met tRNA-METF is mediated by the initiation factor IF2. The F-met begins every polypeptide chain synthesized by E. coli, but it is usually removed after translation is complete. When an in-frame August is encountered during translation elongation, a non-formulated methionine is inserted by a regular MET tRNA MET. After the formation of the initiation complex, the 30S ribosomal subunit is joined by the 50S subunit to form the translation complex. In eukaryotes, a similar initiation complex forms, comprising mRNA, the 40S small ribosomal subunit, eukaryotic IFs, and nucleoside triphosphates, GTP and ATP. The methionine on the charged initiator tRNA, called MET tRNAi, is not formulated. However, MET tRNAi is distinct from other MET tRNAs in that it can bind IFs. Instead of depositing at the Shine-Dalgarno sequence, the eukaryotic initiation complex recognizes the 7-methylguanosine cap at the 5' end of the mRNA. A cap-binding protein, CBP, and several other Fs assist the movement of the ribosome to the 5' cap. Once at the cap, the initiation complex tracks along the mRNA in the 5' to 3' direction, searching for the August start codon. Many eukaryotic mRNAs are translated from the first August, but this is not always the case. According to Kozak's rules, the nucleotides around the August indicate whether it is the correct start codon. Kozak's rules state that the following consensus sequence must appear around the August of vertebrate genes: 5' GCCRCCAUGG3. The R, for purine, indicates a site that can be either A or G, but cannot be C or U. Essentially, the closer the sequence is to this consensus, the higher the efficiency of translation. 15.5 Ribosomes and protein synthesis. 385 Once the appropriate August is identified, the other proteins and CBP dissociate, and the 60S subunit binds to the complex of met-tRNAi, mRNA, and the 40S subunit. This step completes the initiation of translation in eukaryotes. Translation, elongation, and termination in prokaryotes and eukaryotes. The basics of elongation are the same, so we will review elongation from the perspective of E. coli. When the translation complex is formed, the tRNA binding region of the ribosome consists of 3 compartments. The A, aminoacyl, site binds incoming charged aminoacyl tRNAs. The P, peptidyl, site binds charged tRNAs carrying amino acids that have formed peptide bonds with the growing polypeptide chain but have not yet dissociated from their corresponding tRNA. The E, exit, site releases dissociated tRNAs so that they can be recharged with free amino acids. The initiating methional tRNA, however, occupies the P site at the beginning of the elongation phase of translation in both prokaryotes and eukaryotes. During translation elongation, the mRNA template provides tRNA binding specificity. As the ribosome moves along the mRNA, each mRNA codon comes into register, and specific binding with the corresponding charged tRNA anticodon is ensured. If mRNA were not present in the elongation complex, the ribosome would bind tRNAs nonspecifically and randomly. Elongation proceeds with charged tRNAs sequentially entering and leaving the ribosome as each new amino acid is added to the polypeptide chain. Movement of a tRNA from A to P to E site is induced by conformational changes that advance the ribosome by 3 bases in the 3' direction. The energy for each step along the ribosome is donated by elongation factors that hydrolyze GTP. GTP energy is required both for the binding of a new aminoacyl tRNA to the A site and for its translocation to the P site after formation of the peptide bond. Peptide bonds form between the amino group of the amino acid attached to the A site tRNA and the carboxyl group of the amino acid attached to the P site tRNA. The formation of each peptide bond is catalyzed by peptidyl transferase, an RNA-based enzyme that is integrated into the 50S ribosomal subunit. The energy for each peptide bond formation is derived from the high-energy bond linking each amino acid to its tRNA. After peptide bond formation, the A site tRNA that now holds the growing peptide chain moves to the P site, and the P site tRNA that is now empty moves to the E site and is expelled from the ribosome. Figure 15.16. Amazingly, the E. coli translation apparatus takes only 0.05 seconds to add each amino acid, meaning that a 200 amino acid protein can be translated in just 10 seconds. 386 Chapter 15. Genes and Proteins access for free at openstax.org. Visual connection. Figure 15.16. Translation begins when an initiator tRNA anticodon recognizes a start codon on mRNA bound to a small ribosomal subunit. The large ribosomal subunit joins the small subunit, and a second tRNA is recruited. As the mRNA moves relative to the ribosome, successive tRNAs move through the ribosome and the polypeptide chain is formed. Entry of a release factor into the A site terminates translation and the components dissociate. Many antibiotics inhibit bacterial protein synthesis. For example, tetracycline blocks the A site on the bacterial ribosome, and chloramphenicol blocks peptidyl transfer. What specific effect would you expect each of these antibiotics to have on protein synthesis? Tetracycline would directly affect a. tRNA binding to the ribosome, b. ribosome assembly, c. growth of the protein chain. Chloramphenicol would directly affect a. tRNA binding to the ribosome, b. ribosome assembly, c. growth of the protein chain. Termination of translation occurs when a nonsense codon, UAA, UAG, or UGA, is encountered. Upon aligning with the A site, these nonsense codons are recognized by protein release factors that resemble tRNAs. The releasing factors in both prokaryotes and eukaryotes instruct peptidyl transferase to add a water molecule to the carboxyl end of the P site amino acid. This reaction forces the P site amino acid to detach from its tRNA, and the newly made protein is released. The small and large ribosomal subunits dissociate from the mRNA and from each other. They are recruited almost immediately into another translation. 15.5 Ribosomes and protein synthesis. 387 Initiation complex. After many ribosomes have completed translation, the mRNA is degraded so the nucleotides can be reused in another transcription reaction. Protein folding, modification, and targeting. During and after translation, individual amino acids may be chemically modified, signal sequences appended, and the new protein, folded, into a distinct three-dimensional structure as a result of intramolecular interactions. A signal sequence is a short sequence at the amino end of a protein that directs it to a specific cellular compartment. These sequences can be thought of as the protein's train ticket to its ultimate destination, and are recognized by signal recognition proteins that act as conductors. For instance, a specific signal sequence terminus will direct a protein to the mitochondria or chloroplasts, in plants. Once the protein reaches its cellular destination, the signal sequence is usually clipped off. Many proteins fold spontaneously, but some proteins require helper molecules, called chaperones, to prevent them from aggregating during the complicated process of folding. Even if a protein is properly specified by its corresponding mRNA, it could take on a completely dysfunctional shape if abnormal temperature or pH conditions prevent it from folding correctly.